Boy, cool. Everybody coming on in and settled. Amazing. Welcome to the family conference. I'm orienting myself. I only get 15 minutes, and if you guys know me, I talk for longer than 15 minutes, so I probably should get started. Otherwise, I'll never get to our lovely speakers and keynote. So I'm going to go ahead and jump in. Welcome to the Family Conference. I'd like to extend a special thank you to our sponsors and partners for helping make this event even happen. We've had wonderful support in getting this going. This is our fifth PFIC specific conference, I believe, and we can't do it without the support and partnership from our sponsors. See, it takes me a little while to get going. Also want to thank our incredible planning committee. So we had a group of volunteers this year who helped. They met monthly, they fielded emails, they gave us ideas. A lot of the program elements of here was brought to us by our wonderful conference committee. So thank you for your volunteerism. She just entered the room. I have to give mad props to Melissa Kochanowski, who is our incredible Associate Director of Programs. This is her first year spearheading this conference, so the vision and the execution was led by Melissa, so please give her a huge round of applause. And of course, we can't have an event without our wonderful speakers. These speakers volunteer their time. They bring the information to you all. They open themselves up for questions. So we really appreciate all of those who have joined us as speakers. So another round of applause for all of our conference speakers. A reminder, as Tala just told us, the Family Zone is located in Ohio 1 and 2. There's activities there if you want to take a break, if you want to go do a project, if you just need to step out and you need a space. Out the doors and to the left is the Family Zone. And we have a Get Involved booth here with PFIC Network. So there are a lot of opportunities to get involved. Tara, I don't know if you're in the room, but Tara's spearheading the Get Involved booth. It's right out the door. So if you have questions on how you can get involved, big or small, with the PFIC Network, we have opportunities there. I will highlight that we are planning for our upcoming PFIC Awareness Day. And we are taking interviews, short interviews from patients, families, siblings, clinicians, researchers, anybody. So please stop by the booth and talk to Tara about those opportunities. And this is a hybrid event. And so many of you have joined in person, and we have, I think, 50 plus individuals who are joining us online. So for those who are in the room, you can join via Cvent. You should have an email for registering if you have not yet. If you have any questions, you can see Sarah at the registration booth. So go ahead, you can fill out your attendee profile. You can find the agenda in there. So go ahead and utilize that as a tool. And then for our virtual participants, we do encourage an active Q&A through Cvent. We have our wonderful moderator, Dr. Schneider, who is managing the Q&A. Disclaimer, I haven't prepped him at all for questions, but he is willing to field questions, so I had to put that out there. But please engage through Cvent Hub. Also, we do have a wonderful photographer with us. Paul, please raise your hand. Sorry to put you on the spot. I didn't prep you for that either. But Paul will be with us, so it'll be photography. And I will hold you back from lunch to have a group photo, so just prepping you that for now. Cool. And a few disclaimers. So this event is for educational purposes only. You may hear information here that you might want to take back to your own, that is relevant for your own care. Please consult your doctors with everything. We encourage you bringing questions back to your doctors, but please recognize that this is not this is for educational and not consult purposes. Zoom is not a HIPAA-compliant platform, and honestly, this room is not a HIPAA-compliant room. So just recognize that if you're sharing personal stories, names, etc., to please do so at your discretion. They are not protected under HIPAA privacy laws. And then, of course, as mentioned before, the sessions will be recorded and distributed through our Cvent platform, and then posted on the website for educational purposes as well. And then again, as the day goes on, we encourage your questions. We have a few microphones and we can go around the room. Our conferences are pretty open platforms. This is your opportunity to engage with speakers. And honestly, clinicians, researchers, we also encourage you to ask questions of patients. And so this is a bi-directional, a two-way street. So please do not be shy, ask your questions. Oh, I guess I'm supposed to give my introduction now. So I wear many hats in this organization. My name is Emily Ventura. I am executive director and co-founder of PFIC Network, but I'm also a parent to a 14-year-old girl who has BSEP deficiency. Turn my phone on silent. And I'm going to share a little bit about my experience. Because of my different hats, I'm going to take off my executive director jacket, put on my parent hat for a moment. And talk to you a little bit about our journey. So I'm going to take you back to 2012. I'm spending my 27th birthday at Cincinnati Children's Hospital, where my daughter is having her very first liver biopsy. Hospitals are a very familiar place to me, as at this time I was working as a critical care nurse for 6 years. I am definitely that pain in the butt parent who knows way too much about navigating hospitals, asking the nurses about my daughter's flip P waves, telling them that the heat and air conditioning isn't working, so please call maintenance, all of those things. Nurses and doctors really do sometimes make the best and the worst patients. This was our second visit to the hospital in just 4 weeks. Something was going on with my 5-month-old daughter's liver. She had been in liver failure at just 5 months old. What? I know liver failure. I work with liver failure patients. How can my 5-month-old be in liver failure? This was we spent 5 days inpatient at that time. An injection of vitamin K had done the trick, and we were able to leave with a plan to go back for follow-up. We were in some ways grateful to leave, but we still didn't know why. Waking up to the sounds of the city in Cincinnati, Ohio is much different than waking up back home to the birds and cicadas in Kentucky. It is now our very first routine specialist appointment. But really nothing about a pediatric hepatologist appointment for your 5-month-old is routine. But that is the technical term, routine. I knew going into this appointment that Cedar had a diagnosis. I had received a phone call just 5 minutes prior to leaving for that visit. Cedar's genetic tests are back. She has progressive familial intrahepatic cholestasis. Phew, I thought, this is great news. We have a diagnosis. We finally have a reason. We're going to go into this appointment today, and we're going to talk about a plan, and life is going to get back on track. I was optimistic. I was actually hopeful going into that appointment. It was a mix of anxious anticipation, like right before you get a big test score back, with results that can shape your future. That hope fizzled quickly as we sat through that first appointment. This was a very rare disease. The doctor had only seen 2 cases in his career, and they actually could not tell us what to expect. We learned that she would likely suffer from a severe and incessant itch, that she would likely need a transplant before age 10, but there was no way of telling us when. That there were some surgical options that existed that might help her itching, but they didn't know for sure. Clinical trials were coming, but they would not benefit her. They sent us home with off-label medications and a 3-month follow-up where she would need more labs, more tests, all just to monitor and wait. Wait for her liver to fail, maybe. The car ride back to Kentucky was silent. Cedar was asleep in the back. Her dad and I had no words. That 2-hour drive in that moment, which we would take more than 300 times over the course of her young life, was haunted by a hopeless fear of what was going to happen next. We lived that fear in isolation for the next 3 years. We battled countless sleepless nights, changing bloody sheets, without one single person understanding the trauma that we were feeling. We tried the surgical diversion, that failed. Our closest friends and family knew that we were going through hardship, but no one really understood. How do you describe itching without someone offering you a lotion to make it better? How do you let people know that normal blood tests are never actually normal, and that it's hard to celebrate even those normal tests? You're always anticipating the next set of labs that is coming in the next 3 months. How do you tell people that's not okay? Waiting, wondering if every crying spell means that her liver is failing, that every cold, ear infection, or common fall down makes you wonder if it is going to make her liver disease progress. How do I tell people my feelings on liver transplant is the only life-saving option? That it's not as easy as just transplant her and move on? I needed to find someone, anyone, who could understand what I was going through. I began searching online. I had seen a website before and I had tried to connect, but never really got through to anyone. A few years later, I tried again, desperate this time. And thankfully, I was connected to a group on Facebook. I never thought to look on Facebook. Why had I never thought about that? It was right there the whole time. There were 75 people in one Facebook group, and these people got it. They're asking the exact same questions. They're experiencing the exact same things I am. I'm not crazy, and I'm not alone. I'm not the only one who is living with this, watching my daughter live with this, fearing for what's next, waiting impatiently for every next doctor appointment, wondering if that's the one where they are going to tell me that it's time for transplant. I was consumed. I spent 3 hours straight sitting at my computer, scrolling through 8 years of posts from this group of just 75 people, just so that I can take in every single detail of what was being said. I didn't interact right away. I just took it in. But after a few days, I did introduce myself. I shared a little bit about why I was in that group, about my daughter's journey, and I was instantly welcomed by all. It didn't matter what subtype my daughter had, didn't matter how old she was, what procedures she had and had not have, if she was itching or not, if she was transplanted or not. I was welcomed by everyone. That was the very first day I actually felt a glimpse of hope. I was no longer alone. We were no longer consumed by this disease. We were learning to live with it. My entire perspective on being a rare disease parent had shifted in just 3 days of finding a community. It was in that group that I met Robin. Some of you know Robin. Robin had 2 children with PFIC, and she had been working with clinician researcher experts from all over the world. She was flying her children from Texas to Germany to participate in BSEP research. She was working with researchers in a research network called Children. She had also began speaking with the FDA and pharmaceutical companies who were looking at clinical trials for this group of diseases. I didn't know any of that when I first joined the Facebook group. I was just there for support. And I would see her post from time to time about things she was doing, and a few people would jump in and help out. But I thought, oh, speaking with the FDA isn't for me, I'm new. I wouldn't know what to say, but I'd love to do something, just not right now. I'll wait for that next time. Well, one day, next time came. Robin reached out and said she could no longer run the website, pfic.org, and she was looking for someone to help keep it going. The timing was terrible. We were 5 years into our journey, and Cedar had just been listed for transplant. In so many ways, this was the hardest thing that we've done, listing her for transplant. But honestly, in PFIC, it was a little bit of a relief. The choice had been made. It's time for transplant. We're not living with the uncertainty anymore of when that time will be. There's a conflicting sense of nervous anticipation and relief. But for whatever reason it was, as she was listed for transplant, I decided that was the time for me to volunteer. I'll be honest, I had no idea what I was getting myself into. I thought I was signing on to maintain a website, perhaps update it, and be a more active point of contact for people when they need support. I was joined by 2 other mothers, Tara Kearns from Maryland and Melanie Karakaritos from Australia, and the 3 of us just started to figure it out. We would meet on Facebook Messenger and share ideas that would translate into, that sounds great, let's do that. Then we would quickly learn that doing that would take effort that the 3 of us just did not have. We were working full-time, parenting children with a chronic disease, and the mere process of getting paperwork submitted to get a charity going and approved took over 14 months and money that we really didn't have. Everything was new and confusing. We were learning everything from scratch. But we went ahead and we rebuilt the website, pfic.org. We began meeting clinicians and researchers who had specialized in these diseases, and we were leaning on them to volunteer their time to share information so we could update that website. We were meeting the pharmaceutical companies who had clinical trials in place, and at the same time, we were meeting patients and families from all over the world who wanted access to those trials and access to this information. We were bridging gaps, sharing our community's unmet needs, participating in conversations that could help shape the future of discovery, both knowledge and treatment. It was my first real taste of patient advocacy at an organization level. But I still felt a mix of emotions. I was still working full-time and coming home to then work full-time on advocacy while my daughter was recovering from her liver transplant. We were moving the needle, but I was in a constant state of conflict. Expanding the organization's capacity was not just an evolutionary goal. It was an essential next step to ensure the sustainability of the PFIC network and all of the activities that we can support. In 2023, 5 years after our foundation was established, we applied for and received a Chan Zuckerberg Capacity Building Grant, which enabled us to change our organization's trajectory for the better. Through this opportunity, we improved our ability to reach patients and families and ensure that up-to-date and relevant information can be shared on diagnosis in a disease group that is life-altering, but experiencing advancements very quickly. With expanded capacity, we have hosted and co-hosted 5 convenings, established and published a patient registry that includes patient-reported outcome measures, and organized community-wide discussions through these conferences about clinical management needs and the desperate need for new treatments. But most importantly, or of equal importance, it has provided a space for community with diverse backgrounds and needs that come together under a unified identity, the PFIC community. We are all here together in this room. We're not the doctor, the patient, the parent, the sibling, the grandparent, the researcher, the industry member, the friend. We are all a group of people who are together under one very important mission: to improve the lives of patients and families affected worldwide by PFIC. I'm a little bit over time, but I want to end with a very quick reflection back on my own story. Cedar is 14 years old now, and she is thriving. She's attending high school next year and keeping me very busy by being an aerial arts performer and playing soccer. However, her journey after transplant has not been simple. Cedar's specific BSEP mutation puts her at risk for a post-transplant complication called alloimmune BSEP deficiency. In the body, her rejects the BSEP protein that comes with her new liver. On the surface, she's experienced multiple episodes of rejection and a full relapse of her disease in the 9 years that she's been transplanted. Her transplant was right at a time where this was only being discovered and published in a handful of cases. And it was Robin, the PFIC mom who was taking her kids to Germany from Texas for this study, who were among the first diagnosed with this phenomenon. When Cedar had her first rejection 2 months post-transplant, and I was working with Robin on the foundation, she said, "Emily, you should ask your doctors about this anti-BSEP work." So I did. Cedar's doctor said, "Sure, that's a possibility, but we don't have the ability to test that yet. We don't have the lab." And I said, "I know who does, and here's the doctor's name for Germany." And he contacted them, and he sent her tissue to Germany. And so Cedar was diagnosed with this very rare complication post-transplant, I know, on diagnosis, because of that connection and because of this network. So don't underestimate the power of these connections. And please don't not ask the questions. Thank you. Ron, apparently this is your slide. Good. So I'm going to invite to the stage a friend, a colleague, and the PFIC Network Medical Advisory Board Chair, Dr. Ron Sokol, who is going to take us through the evolution of PFIC and all of its journey. So here you go, Ron.
Well, thank you, Emily. It's an honor to be asked to present to all of you. And it's I'm in an awkward position having to follow Emily with that very emotional story of hers. But let's move on, and I ask you all to let me know if I'm hitting the mark here and talking about the evolution of PFIC and tomorrow's hopeful solutions. Just some disclosures that I consult for number of companies, most of them involved in searching for new treatments for cholestatic liver diseases, Scientific Medical Advisory Board here, and I receive NIH grant support for a number of studies that I do. So, first of all, what's in a name? How in the world did this disease get a name that's almost impossible to pronounce? And because of that, we all call it PFIC or PFIC. How did this happen? How did it evolve? First of all, one should know that PFIC is not a single disease. It's what we call in medical terms a phenotype. It's an observation of clinical symptoms that all fit together in one bundle that allows us to say this child has PFIC. At least that's the way it used to be until it evolved into a genetic diagnosis. So first, let's talk about what it actually means, and let's start at the back. So the C stands for cholestasis, which means impaired or poor bile flow. But what actually is bile? So bile is the yellow liquid made in liver cells that's pushed out of the liver cell into these tubes called bile ducts and into the intestine for a variety of reasons. The components of bile tell you what it does. So first of all, bile contains bile acids, which are detergents, among other things, help digest fat so that the fat that we eat in our diet can get into our intestines, to our liver, and to the rest of our body. Bile also is the main way that the body gets rid of various waste products besides urine. And these include various toxins in the environment, drug metabolites, bilirubin, cholesterol, and another lipid called phospholipids. Finally, bile, and I'm not going to really dwell on this, but the components of bile really do influence the bacteria that normally live within our intestines and our colon, called the microbiome or microbiota. And when there's good bile flow, certain bacteria live there. If there's impaired bile flow, other bacteria live there. And there's a whole field of research now looking at the interactions of the bacteria within our intestines and the liver. It's called the gut-liver axis. Unfortunately, we don't have any new treatments yet based on the knowledge that's been obtained, but it's definitely an area that many are doing research in. So once again, the bile is made in the liver cells, goes through those tubes, the bile ducts, into the intestine. So cholestasis really is the impairment of bile flow, which prevents the buildup of, again, various toxins, including bile acids, in the liver. And what do bile acids do when they build up in the liver? Well, they cause soreness or inflammation, swelling of liver cells, eventually scarring in the liver, and they can eventually then lead to cirrhosis. The buildup of bile acids also is related to the itching that we just heard of from Emily, or the medical term is pruritus. The itching is probably not caused directly by the bile acids, but something related to the bile acids that, again, nobody's quite sure about, but we do know that if you can lower bile acids in cholestasis, generally the itching does get better. And then finally, bilirubin is one of the components of bile. And if the bilirubin accumulates, this is what happens. In adults, frequently the skin gets very yellow, as well as the whites of the eyes. But in infants, sometimes it's much harder to see a mild degree of cholestasis. The white part of your eyes, the sclera, do get yellow generally before the skin does, because the white background and the thin membrane over the sclera, as you can see in this baby. So the I stands for intrahepatic. And there's two major kinds of cholestasis, or impaired bile flow. And one is the type that occurs inside the liver. Intrahepatic actually means in the liver. And this happens in the liver cells themselves, or the very small bile ducts and the what's called canaliculi in the liver. And this is opposed to blockage in the extrahepatic bile ducts. And this is the disease biliary atresia in children, gallstones in adults. And they're really treated quite differently, although clinically can look identical in an infant until we do testing. The F stands for familial. And this is due to the fact that it's we now know these are genetic disorders, and because of this, it can recur in families. Originally, we thought roughly 20% of children with these types of presentations clinically had a familial form of a disease. 30 years ago, we didn't know what caused it, but it was hereditary in nature in roughly 1/5 of cases of infants that we saw with cholestasis. And as we'll mention in a second, now there's at least 13 different genes among our thousands of genes in our bodies that have been associated with cholestasis in infancy. Most of these that cause PFIC are autosomal recessive, meaning that one needs 2 copies of an abnormal or variant gene. We used to call them mutations. You need 2 copies for the gene to be expressed as a disease. 1 copy, you're a carrier, which in most cases causes no problems whatsoever. So parents are frequently perfectly fine, yet they'll have a child with PFIC. And BSEP and FIC1 are 2 proteins that, again, present almost always autosomal recessively. However, there are some genes, such as MDR3, which is PFIC type 3, that actually can have a disease expression with only 1 copy of the gene present. So it's called a heterozygote or a het. Sometimes BSEP does this, but again, those are the minority of people who are affected. And then the P stands for progressive. In this case, this is not a political term, but it means that the disease increases in severity over time, or medically, we say that this is progressing. Some children with cholestasis get better on their own, and we call it a transient disease. This occurs with some babies who develop cholestatic jaundice. But PFIC rarely resolves on its own, although each child's journey is different, and some progress more rapidly, some progress more slowly. And then there's some very mild versions of PFIC that actually can present in adults. And it's probably not even proper to call them PFIC, although I think some call them adult onset PFIC, but it's basically they have milder changes in the same genes that cause PFIC, but they're so mild that they don't show symptoms until they get pregnant or go on birth control pills or get another drug that might in some way cause cholestasis. And there's several conditions in adults. They used to be called benign recurrent intrahepatic cholestasis, or BRIC, and that term now is used less, but it's caused by the same genes that cause PFIC. So how did this all evolve? Well, back in the 1960s, led by Bob Clayton, who is a geneticist at Johns Hopkins, he studied a group of children that were in the Amish community, mostly from Pennsylvania, who all were descendants of Jacob Byler, who was an Amish leader in the 1850s. And they published this paper in 1969, the first description of this then fatal, because there was no liver transplants back then, intrahepatic cholestasis in an Amish kindred. And the clinical description is exactly the same clinical description that we have today. And it was those children had intense itching, they had growth failure, they had progressive liver disease, they eventually developed cirrhosis, And again, there were no liver transplants, so many of them succumbed to their disease. And that's actually a picture of Bob that I found on the internet with the Muppets, which I thought was really quite cute. The significance of this paper was that he had actually identified what appeared to be a hereditary or genetic cause of cholestatic liver disease. And up until that time, most thought that cholestasis in infants was caused by infections or bile duct obstruction, the disease called biliary atresia. And then a few years later, a group from Toronto described something that was called Byler's bile. And on the right, you see a normal canaliculus, that tiny little tube that I mentioned between liver cells, and it's in the middle with those little knobs. Those are called villi, microvilli, that extend into the canaliculus. Those are two liver cells on either side of it. On the left is Byler's bile, and you can see on the left side of that image there, these granules that don't occur in normal bile. And it was this granular looking bile on, these are electron micrographs or thousands of magnification of the liver. That granular bile became a characteristic feature that actually many of us use to diagnose Byler's disease or Byler's-like diseases if the child had a liver biopsy done. This also was very significant because it pointed to the bile itself as being the cause of this disease compared to other cholestatic liver diseases. In the 1970s and 1980s, after these initial publications in Amish children, others recognized similar cases of children with cholestasis who seemed to have a similar disease but had no relationship to the Amish community. And this was then labeled as Byler's syndrome instead of Byler's disease because there were many similar features, but again, no direct genetic relationship. Also in the 1970s and 80s, it became apparent that children with Byler's disease in similar presentations had low levels of a chemical in the blood called GGT. And this is one of those liver enzymes that we commonly measure in any patient with cholestatic liver disease. It's found in the bile ducts and canaliculi, excuse me, and is almost always elevated. However, in these cases, it was almost always, almost always normal or even lower than normal, which was very unusual, but was a characteristic that many used to label the patient as actually having PFIC. So that was, again, another hint that something was going on in the secretion of bile in this disease. And then in 1988, Peter Whittington and his brother Gene, who then were in Memphis, and later Peter moved to Chicago, they published a paper that was really a landmark paper. They thought that because the Byler's bile appeared to be involved in this disease, and we knew that itching was associated with elevated bile acids, what if in some way you could get rid of the bile from the body surgically and see if itching and other features of the disease improved? So on the lower left, you can see that what they devised, and neither of them are surgeons, by the way, what they devised was working with their surgery colleagues, a way to take a little piece of intestine, sew it to the gallbladder, and then move it to the skin surface so that bile would flow, instead of from the gallbladder and bile ducts into the intestine, flow from the gallbladder to the skin, and the patients would wear a little stoma bag and collect the bile and then discard it throughout the day. So this was called a partial biliary diversion, and you could see the picture there. And lo and behold, in their first paper, 4 of the patients had what they called progressive intrahepatic cholestasis. They left the familial out. But these 4 patients who had surgery at 3 to 9 years of age had amazing reduction in their itching or pruritus. And they had a 4-point scale where 4 was the worst itching you could imagine and 0 was none. And all 4 patients, their itching completely resolved. This was also tried in Alagille syndrome, another cholestatic disease. The results were not quite as good, but in PFIC, at least these initial 4 patients. So this again led to the concept that if you could get bile and bile acids out of the body, that you could improve the condition of these patients. And since then, there's been papers showing that in some of these patients, scarring in their liver also improved over the years after a partial biliary diversion. And then in 1998, everything changed. Why did it change? Well, landmark publications by some of the scientists in the audience here. And the first was the publication by Laura Bull. Laura, raise your hand. Where did I see you? Dr. Laura Bull and many others who studied this at the University of California, San Francisco, and they identified the first PFIC gene called FIC1. That is the gene that, the proteins FIC1, the genes ATP, 8B1. This is the gene that caused PFIC type 1 and was actually the gene that caused bilar disease in the Amish children. This paper caused a whole revolution in the whole field, but it wasn't the only one in 1998. In Richard Thompson's group, with Sandra Strautniks being the lead author and Laura Bull. Richard, you raise your hand, please. There's Richard. They published this paper several months later, which identified BSEP, or PFIC type 2. And this was not all that happened in 1998. The group from France, led by Emmanuel Jacquemin and others from Basset in Paris, published this paper in 1998, and they identified that mutations in MDR3 cause progressive familial intrahepatic cholestasis. And after the term was used in these 3 papers, all of us adopted PFIC now to describe this group of diseases, and that what we all needed was a way to make these genetic diagnoses, because these tests were not available clinically in 1998, and it actually took quite a few years for the revolution in genetic testing to happen that now allows us to make the genetic diagnosis very simply, whereas 20 years ago it was almost impossible. So what about those 3 proteins? What do they have in common? Why would they be involved to cause these diseases? And you can see on the left that the canaliculus is that open space where 2 liver cells, microscopic liver cells, connect. In the canaliculus, there's a membrane that separates the liver cell, the hepatocyte, from the canaliculus. And in this membrane is where these 3 proteins live. And each of them is involved in the transport of bile acids, or those phospholipids that I mentioned, into the canaliculus that connects to the bile ducts, eventually to the intestines. And this is how bile is secreted and how bile acids are emptied out of the liver cell to help with digestion, but also to protect the liver cell. So this was the new biology that we learned from these genetic discoveries. And it's only through these type of discoveries that we then can start thinking of developing therapies that could target these type of areas. So what's happened in the last 25 or so years? Well, a lot has happened. First of all, the human genome was sequenced. We now know there's about 20,000 human genes, and it's actually quite easy nowadays to get all of these measured within a few days if one has the right laboratory that will work with you, and to be able to analyze hundreds of thousands of bits of data, which is not straightforward. But these various technological advances have really allowed us to do this, and the computational advances, including artificial intelligence nowadays, have allowed us, again, to analyze these massive databases from a single patient, and not only make diagnoses, but discover new genes. And that's now led to this long list. Under that line, you could see in 10 years, there were another 10 genes that have been labeled as PFIC genes. Many of these genes are completely unrelated to other genes, but the common thread is they cause cholestasis, they cause itching, they present as infants, and almost all of them are progressive, and many lead to the need for liver transplantation. So what do they all have in common? Well, again, they're all involved in one way or another in the biology of bile secretion or protection of the liver cell from bile acids, which, as I said, in high concentrations are detergents and can damage cells. Now, rather than have a list that goes on forever of type 13, 14, 15, 16, others, including Dr. Thompson, have proposed that we should actually look at these as individual diseases, but we could break them into certain buckets or categories based on what the function is of the proteins involved in each of those genetic diseases. So for instance, many of these diseases, I just showed you 3, involve the transport of molecules through the liver cell into bile. BSEP, MDR3, et cetera, are examples. Some involve stabilizing that canalicular membrane, such as FIC1. Others are proteins that code for other are genes that code for proteins that form what's called tight junctions. These are the way the liver cells connect to each other to prevent backflow of bile into the liver cells. There's nuclear signaling, genes that control the expression of all of these other genes, and finally, trafficking of vesicles within the liver cell. A lot of transport of these proteins to the cell membrane is through these tiny little compartments called vesicles. Now, some of these genes are expressed in other tissues and involve other tissues besides the liver, and you'll hear more about that today. So every one of these genetic diseases has its nuances, and some of them, as we heard from Emily, can lead to a recurrence of sorts after liver transplant. Many of them are completely cured by liver transplant, but once again, every child who's affected has its own story, and we have to approach each child and each adult who's affected with personalized approaches. So the significance of these proteins in identifying the underlying cause is that each one could potentially be a target for a therapy for that individual disease. Now, currently, the way we treat cholestatic liver diseases, we treat them all pretty much the same. And that includes diseases like biliary atresia, Alagille syndrome, and the PFICs. And we have this list of therapies listed there that are commonly used. There's therapies that are in development, though, based more on the biology of those proteins. And when this slide was made, actually, IBAT inhibitors had not yet been FDA approved. They were originally called ASBT inhibitors, more on that in a second. But really what we'd like to do is move from that to a more personalized approach where we have therapies that are disease-specific, gene-specific, and eventually personalized to the variants or mutations in an individual patient. And that really is the goal for almost all rare diseases. So one example, and the most influential example, has to do with understanding the biology of the enterohepatic circulation of bile acids. So the enterohepatic circulation involves bile acids, the little green blobs coming from the liver down the bile duct into the intestine, and then getting reabsorbed and the end of the intestine, called the terminal ileum, by this protein, IBAT, that transports them into the blood back to the liver. And it just recirculates. And this is a way that the body, if you will, is a little bit more efficient and not making bile acids that just dump out of the body, but it reuses the bile acids. And this shows it, and thank you, Richard Thompson, on a cellular level. That's two hepatocytes on top with the canaliculus. Through BSEP, the bile acids in green get transported into bile, go into the intestine, get reabsorbed by that intestinal cell on the bottom through ASBT, which is IBAT, and then go back to the portal blood to the liver, get taken up by the hepatocyte, by NTCP, another transporter, and round and round. In PFIC2, it's BSEP that malfunctions, and because of that, bile acids will build up in the liver cell and less bile acids will get into the intestine. So the theory is, what if you block the reuptake of those bile acids through the intestine? Then they won't go into the portal vein, won't go back to the liver. Can you eventually reduce the buildup of bile acids in the blood and in the liver? And this is really the base, and then the bile acids would remain in the intestine and eventually end up in the toilet, which, as far as we know, do not harm the porcelain in your toilets. So this is the theory behind that, and as I think most of you know, that these medications have been developed. There's two that are FDA approved for using PFIC, maralixibat, that's produced by Mirum, and odevixibat, that's produced by Ipsen. These are effective in quite a few patients. What they do is they reduce the serum bile acids, as we mentioned. They frequently reduce itching quite a bit, sometimes to zero. They improve growth in Alagille syndrome, and now there's some data showing improved growth in PFIC. They can improve survival because if your transplant is because of itching, and your itching is better, then a lot of those children will delay or maybe not even need a liver transplant. The side effects are minimal. It's oral doses, and occasionally there's elevation of liver blood tests, but those are usually manageable. The issue is that only 40% at most of children with PFIC respond. And why? It may be that the proteins are so damaged by their genetic mutation that not enough bile acids get into the intestine for this to have an effect. So there is still a large unmet need for those patients who respond to these drugs. They're wonderful. For those that don't respond or aren't even candidates for these drugs, we need new therapies, plain and simple. So the future therapeutics for cholestatic liver disease really involve targeting these bile proteins that I just mentioned with small molecules. Small molecules are basically pills that you can take. The IBAT inhibitors are small molecules, or use technology to add RNA back into our bodies to produce those proteins, get the RNA to the liver, and at least temporarily produce the missing protein. I think you're all familiar with the RNA vaccines for COVID. This is a little bit different. This isn't to vaccinate somebody against the proteins, but it's using similar technology to get messenger RNA into liver cells. And ultimately, gene therapy or gene editing to either replace the damaged gene or to edit it and take out the bad gene and put in a good gene. And these are the major categories of where the pharmaceutical industry and biotechnology is headed in terms of developing therapies for genetic diseases. So this just shows you these pink bubbles are a whole bunch of drugs that are either in development or in clinical trials to target the liver cell, the bile duct cells called cholangiocytes and the intestinal cell that are involved in the enterohepatic circulation of bile acids. But there's challenges with every rare disease, and PFICs are no different. And that is that each is a very rare disease. No single medical center group of investigators can make a whole lot of progress without a lot of collaborations. And now with our new technology and understanding of genetics, there's really no limit to discovery of causes of genes and diseases and the developing of novel therapeutics. But to do this, we really all need to work together. And this includes universities, government agencies, which currently have issues in the United States, which hopefully will be resolved soon in terms of funding for research. But our hospitals, our pharma partners in industry, and probably most importantly, patients and families and patient advocacy groups, because we clearly can't discover the cause of the disease or do clinical trials and test new therapies without people who are affected by the disease. So, excuse me, that's my warning. Emily's saying you're over. But we really need everybody's collaboration so that we can discover the causes, translate these into therapies, and improve clinical outcomes, and hopefully avoid need for liver transplant. That's the goal. So there's at least 3 different networks that are working on this. We'll hear more about NAPID, which Hank-John Verkade is here, who runs that. Hank-John, raise your hand. He's back there. The Childhood Liver Disease Research Network that many of you are perhaps participants in our large study called LOGIC. We thank you. It's present in 12 centers in the United States. And now PFIC Network is going to begin projects that will also help with this collaboration. So I want to thank you for your attention. I'll be here all day. If any of you have any questions for me, please stop by and say hello, and I will turn it back over to Emily. Thank you.
Thank you, Ron. And it wasn't me that gave you the warning. That was something else, but worked out. So yes, thank you very much. I'm going to go ahead and transition us and invite our presenting sponsors on stage. So Ron had alluded to the IBAT inhibitors as part of our evolution, and we're very grateful to the development and to the companies who have provided those and continue to pursue access for our patients and families. Both have been highly supportive and wonderful partners to PFIC Network throughout the years, both Mirum and Ipsen, and so we'd like to give them an opportunity to share a little bit about their work. So I invite Sam to the stage with Mirum Pharmaceuticals. Thank you, Sam.
All right, hi everyone. I'm Sam from Mirum. I lead our patient marketing here. First, just want to say a huge thank you to Emily and the entire PFIC network for allowing us to be here. We don't take it for granted at all, just the honor to be in the room and hear from all of your stories. I do want to welcome you to come out to our table right outside these doors, learn more about Mirum and what we do. You can play a fun game of Plinko, earn some prizes, and just ask questions. We're here to answer. And what I would like to do, rather than you all listen to me, I'd love to invite up Stuart, who is actually an adult living with PFIC, so he can share his story and a bit about his diagnostic journey. Please join me in welcoming Stuart.
All right, well, thank you all for the nice reception and everything. I'd really like to thank Emily. She invited me here about 3 or 4 months ago to come share my story. My story started back about 45 years ago. I was diagnosed when I was 16 years old. Came home from a family vacation. We were sitting there at my grandma's house and everything, and I just got this little itch that just started itching and it wouldn't go away. And it wasn't much the first day, but it was just a little annoying one that would just come back. You'd scratch it, it'd feel good for a little bit, and then it'd come back again and everything. So the itching gradually progressed and started getting worse as we went. It was just a matter of weeks, and then you get to the point where you're no longer sleeping anymore and everything. So that's the biggest thing. So it got severe enough to where I ended up being hospitalized. This is back in 1980. I was hospitalized in intensive care for about 30 days and everything. And eventually they ended up having to do a liver exploratory on me because they gave no options. My bilirubin had climbed to 36 and they were kind of out of things to do. While they did that, obviously I just saw some work up there that related to it. They ended up putting bile duct drainage tubes in. And so when they released me to go home, I had the bag that came out of my side. I had 2 ports on it, and it drained the bile out of there. So when I got to go back to school, I had the joy of having to have my mom come up to high school and drain my bag for me after lunchtime, and then I'd do it again later on that evening. So that's kind of the interesting thing is I start to see more and more doctors speak about this. I start pinpointing all the things along my journey. But for decades, I experienced flares every few years. In the late 1990s, we found a combination of drugs, the rivampin, eudicerol, and phenobarbital, that kind of helped solve the problem a little bit for me. It would usually take about 2 or 3 days of dosing and everything, then my bilirubin and everything would start to drop again for me. But the one thing I can tell you is there's never a clue on what caused it. There was never a thing that I'm walking along, it just starts. The itching is the thing that I notice first that it starts with. Over the years, learned how to function through all the discomfort. My wife and I raised 3 children together. I held a steady job as a manager at a large hardware store, so kept me very busy, mind occupied. And a mind occupied, it lets you move forward and forget about the things that are going on in the background. My journey became even more challenging after my longtime physician retired in 2008. After that, we struggled to find providers that could actually help me. I know my wife she would actually call people and ask them, hey, this is my husband, we got this case, and can you help us out? And they literally had no clue of what it was. So whenever I'd go to a new doctor's appointment or anything, I have to explain to them what I have or what I think it was, which we told it was bricks. So that's what we told everybody. Well, I have bricks. This is what it is. There is no cure. I just need these 3 medications prescribed for me. When I call your office, I need you to go ahead and put that order in. Don't say we got to run tests, because if you delay this a week, I'm going to be super miserable. So I was diagnosed in 2017 with dermatomyositis, which is an autoimmune disease, which complicated things for us a little bit scared to death of the unknown. That's another one of those ones that's very rare and people don't know about. And so the question was, how's it going to interact with my PFIC diagnosis? Okay. In 2022, everything changed. I started, instead of my little flares lasting for a couple of months, I flared from 2022 to 2025, or into 24, I was in a constant flare of high bilirubin counts. All your liver enzymes were elevated, bile salts were elevated, and everything. And so we ended up finding a new doctor, and was working with her at the University of Medical Southwest in Dallas, Texas, in 2023, and she asked if we had done any genetic testing. We said, no, we haven't done it. Everybody's been telling us there's no cure. They don't know what I got other than I have the elevated liver counts and everything. So got the test results back. Now, it took a while. It took a while because they lost him for a time being at the hospital and everything, and it finally came back. We had it, and I was diagnosed with PFIC-1. So 43 years later, we now have a diagnosis. Okay, so that was in the first of 2024 that we got my diagnosis back on that. In 2024, we had the diagnosis, so it gave us a couple of things by having that diagnosis. It gave my doctors, my new doctor, the ability to have something to go shoot to look at, and this is what we can treat now. We know what it is and everything. And then my wife, she found online information about the IBAT inhibitors through a PFIC patient group on the internet and everything. So we went back and we talked to my doctor about that, and we got the ball rolling on that. So I started taking it. Within weeks of starting the IBAT inhibitors, I noticed meaningful improvement. My itching decreased, my sleeping became better, my appetite came back, I was less irritable. It made probably the single biggest difference in my life. I've not had any concerns about taking the IBAT inhibitor since I've had it. Just remember, everyone's going to have a different reaction. Your doctors are the ones who are going to follow you up with you. If you notice there's something wrong or doesn't feel right, get with your doctor and let them know. But the other thing I will say is be confident, voice your opinions with your doctors, because sometimes you tell them it's an itch and they have no idea what you mean, and you got to make them understand. Some of us know. I met a lady yesterday, and she had the exact same reactions that I had, and so it was nice to see. Yesterday was the first time in my life I've met somebody else with PFIC. So at one point, after things were going along good, one point in time, I quit taking my IBAT inhibitor for some stupid reason, and it took a matter of about a week and a half before I started noticing that the itch was coming back. And you know what that itch is? It's automatic. And so I got back on the IBAT inhibitor, and once again, a week or two later, everything smooths back out, and all my stuff rolling right. So what I can tell you is, been a year and a half now with no flare-up. Again, all my liver tests are normal, and that. So that is the good news. Today, I still live with PFIC, obviously, because there's not a cure just yet, but monitor my health closely. But I'm no longer worried about what happens if I get another flare, because I know there's a treatment out there that directly impacts the pruritus, which for me is the worst thing in the world. Okay, so if they got a cure for that, that's good for me and everything. And with that, it's my conclusion. It's been a long journey, but there is hope out there. I am 62 years old now. I've had this for 45 years, and I've had ups and I've had downs, but it always gets better. And now that we have more and more doctors looking at this and everything and seeing about the gene sequencing, I think the doctor who discovered my genetic gene is here. Yep, there you go. So thank you very much. I talked yesterday, and he told me that you were here, so I thought I'd say hi.
Thanks for being here. Thank you very much, Stuart, for sharing your story. And I'm going to invite Alicia and Raphael on stage to talk to us about Ipsen and your journey in IBAT inhibitors and whatever you'd like to share with us. So thank you for joining us.
Thank you, Emily. I want to thank the PFIC network for having us here today. And I think that I'll focus my thank you to all of you, the parents and families, because I know that you have made a great effort to be here, and it's not an easy task to travel and meet some of your fellow patients that might be living with PFIC. We don't take this opportunity lightly. We are here to listen and to learn from you, because you're the true experts of what it really means to live with PFIC. I'll introduce myself. My name is Rafael Sierra. I'm with Ipsen. I'm the US medical lead for the rare disease portfolio. And this is my colleague, Alicia.
Hi, everyone. I'm Alicia Mavis. I'm actually a pediatric hepatologist that recently joined Ipsen in the rare disease as a medical director.
Thank you, Alicia. And I would like to just tell you in a very simple line what we do at Ipsen. Our main mission is to be able to bring therapies and medicines to patients that are living with rare and very complex diseases. We started one of the journeys back in 2023 by bringing an FDA-approved therapy to treat itch produced by PFIC. This inspiration to work with the liver disease community has now evolved to have a team of many researchers, many internal hepatologists, and we're now looking into 5 different liver conditions. But none of this progress comes without a true purpose. Your voice, your advocacy, advocating for other patients, participating in clinical trials is so critical. At Ipsen, we like to listen to your stories. We like to listen to hear what you're living through. Last evening, yesterday, we had the opportunity to sit down with a couple of families to listen. What is it like to live on a daily basis with PFIC? We also asked questions. What are the challenges when some of your kids are transitioning to adult care? I think that is very important for us. And all of the materials that we do are reviewed by patients just like yourselves. Why? Because we want to make sure that everything is used and it's relatable, because there's a real story behind it, not for a piece to be put on a shelf. With that, I just want to leave you a little bit with a thought and a promise that it is not PFIC that defines you. It is actually the journey, the experience that you have, the ones that guide our work at Ipsen to make our lives and your lives better. Alicia?
Thank you, Rafael. Again, I just want to echo what he said and say thank you for inviting us and allowing us to be here. That message that he just said is so important. I've been able to see firsthand how PFIC really can affect children and adults alike. And so those of you that are representing the community here today, it is just so important. I'm going to keep it brief as I see Emily's looking at me. I've worked with her for a few years now in other areas, and I've also managed different programs like this. So I just want to say again, community events like this are so, so important, and it's how we learn and how a company like Ipsen can continue to go back and talk to our team. So please continue sharing your stories. Please continue advocating and working through this. Every story we do take back to our teams to try to identify problems that we can help solve. So I just want to close in thanking you so much for the privilege to even be here and be joining your community event. And if you have any questions about Ipsen or about what we do or what we've shared today, please swing by our booth and talk to our colleagues. We have John and Anna running the booth, and I think there's even some goodies there. But to everyone in the room, whether we've met before today or just meeting for the first time, we really do thank you and hope you have a wonderful day and a wonderful educational event today.